Hey guys. We're going to learn everything there is to know about cells in just 10 minutes. Well, let's get this started. So what are cells? Cells are just the basic unit of all living matter. And this idea is known as the cell theory, which is about cells being the building blocks of living matter, smallest unit of life, and they're made up of pre-existing cells by division. Some types of division are binary fission, meiosis and biphosis. Cells also contain information for growth, development and behavior, plus the site for all chemical reactions of life. And the study of cells is called cell biology. These are some examples of different kinds of cells in their varied shapes, like red blood cells are round and biconcave. Nerve cells are long and branched. This, here is a typical cell, that is all cells have a cytoplasm enclosed within a membrane containing proteins and nucleic acids. Next, to classify cells. Organisms are organized into two groups, as you can see, unicellular and multicellular. Unicellular are organisms that are single-celled, and multicellular are organisms that have many cells. Now cells are of two types, eukaryotes, which contain membrane-bound organelles, and prokaryotes do not have these organelles and also lack true nucleus. Prokaryotes always have a nucleus. Some examples of prokaryotes are bacteria like E. coli and archaea. Archaea are just species that live in extreme conditions like super salty water called halophiles. We are going to focus on the cell structures and its functions of animals, plants and bacteria in the most fun way possible. Also, some multicellular organisms are complex, having an organization where cells form tissues and tissues form organs, like in mammals. As you can see, here's an easy overview of what's absent and present in animals, plants and bacteria. The structures, organelles, are part of a cell and are adapted to carry out different functions. And this region here is grouped to form the endomembrane system, since their functions are coordinated. Bacteria lacks organelles which are present in animals and plants. Animals obviously lack plant organelles. For the structural view, this is a prokaryote of bacteria. And this is the cell wall, the vacuole for the plant cell. And this side is the animal cell lacking the cell wall. Other than that, animals and plants almost share the same organelles. Firstly, let's look at cytoplasm and cell membrane, which are present in all animals, plants and bacteria. So this is the cytoplasm. And this aqueous fluid part in it is the cytosol. The insoluble suspended particles are just ribosomes and other organelles. As for the function, most enzyme-controlled chemical reactions takes place here in the cytoplasm. Like those associated with cell growth, protein synthesis, like the amino acid activation stage, and so on. Secondly, the cell surface membrane is portrayed as the fluid mosaic model that surrounds the cytoplasm, retaining the cytosol. The cell membrane also consists of different proteins aiding in cell signaling. It is semi-permeable and is responsible for the movements of different molecules in and out of cells through passive transport, or water through osmosis, and iron through active transport. These glycolipids and glycoproteins, together known as glycocalyx. It functions as receptor sites for chemical signals, cell recognition, and binding cells to form tissues. These filaments of cytoskeleton helps organize and maintain cell shape. They also anchor organelles together. Next, the nucleus is present in animals and plants. This here is the nucleus, and this dark spherical body is the nucleolus, the site where ribosomes are synthesized. Here's the nuclear envelope, which contains pores. Pores enable speedy movement of molecules between nucleus and cytoplasm, and vice versa. The nucleus, which controls cell activities, contains chromosomes. Chromosomes are thread-like structures, made of DNA, visible during cell division. And it's passed on to new individuals when sex cells fuse, that is during sexual reproduction. This is the nuclear matrix or nuclear plasm, which contains the nucleolus and chromatin. DNA exists in the chromatin form in the nucleus before cell division. The structure of chromosomes site where almost all DNA replication and RNA synthesizes occur. These are centromeres, and chromosomes are classified according to the position of the centromere. You can see the DNA replication process taking place here. Now the mitochondria is present in animals and plants. Sausage-shaped structure, the site for stages in aerobic respiration to occur and for ATP synthesis. ATP itself is just a small organic nucleotide. It is the source of free energy, taking part in energy-requiring reactions like muscle contraction and energy-yielding reactions like aerobic cell respiration. Mitochondria is a double membrane. This is the outer membrane, a smooth boundary, and is permeable. The inner membrane, which forms enfoldings, called crystae, increasing surface area. This runs towards the matrix, the interior of the mitochondria. The region which contains enzymes, circular lengths of DNA as such. Mitochondria contain 70s ribosomes. It is the powerhouse of cells, all right then. Ribosomes. Tiny, dark dot-like organelles. They are found in animals, plants and bacteria. The size of ribosomes in mitochondria, chloroplasts, bacteria are smaller, 70s only when compared to ribosomes in the cytoplasm and endoplasmic reticulum, which are 80s. And S is nothing but called Svedberg unit. Just the unit for tiny structures. It is the site for protein synthesis, like in transcription and translation stages. Cell proteins formed, each have a structural role. Example, collagen, having great tensile strength, is a great component in our skin and bone. Other cell proteins act as biological catalysts. Now we move into endoplasmic reticulum. This consists of two parts. This is the rough endoplasmic reticulum, having ribosomes. And this is the smooth endoplasmic reticulum, does not have ribosomes. Also, the endoplasmic reticulum buds off from the outer membrane of the nuclear envelope. So here is the site for protein synthesis. And SER manufactures lipids, steroids, reproductive hormones and such. Next, the Golgi apparatus is found only in animals and plants. This region here with its vesicles. Its main function is to pack and deliver materials, like proteins and lipids, in the form of vesicles. Then, a number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae. This curvy edge region. Also specific biochemicals, like hormones, are synthesized here too. The vesicles that chip off the Golgi apparatus form lysosomes in animals. And in plants, it is used for cell wall formation. Now, lysosomes are tiny spherical vesicles, having a single membrane. They contain digestive enzymes like lipases, protease and so on, which are called hydrolytic enzymes that digests food materials containing proteins, lipids, etc. They also destroy damaged organelles and engulf bacteria or viruses. This is done by the fusion of lysosomes with the vacuoles in which materials are taken in. And when an organism dies, hydrolytic enzymes cause self-digestion by escaping into the cytoplasm. This process is known as autolysis. Now, vacuoles. They are small and temporary vacuoles are found in animals for storage. Large and permanent vacuoles are found in plants. This is the plant cell vacuole. It gives a supportive hold as it occupies 90% of the cell's volume and it functions as a storage too. Then, microtubules and centrioles. Firstly, centrioles are found only in animal cells. It contributes its role in cell division. Here's an easy way to look at this. There's an organelle called centrosome which produces microtubules. And this centrosome is made up of two centrioles. Centrioles are tiny organelles that consist of 9 paired microtubules. And these centrioles separate during cell division stages, which is nothing but IPMATC, a simple way to remember. During these stages, centrioles play their role. They grow spindle fibers, and these spindle fibers aid the movement of chromosomes in the division process. Microtubules, they are found in both plants and animals. They guide and direct organelles within cytoplasm. They also shape and support the cytoplasm. They are made of a globular protein called tubulin. Lastly for this, just remember that animals have a single centrosome which is also found in fungi and algae too. Next, the cell wall, which is found in plants and bacteria. And plasmid are found only in plants which acts as a connective medium. Bacterial cell wall is made of peptidoglycan and plant cell wall are made of cellulose. Overall, cell walls function to support, enclose and protect the cell. It's also permeable to all materials. It prevents damage because of its rigidity and counteracts osmotic pressure. Animals do not have a cell wall and it's a disadvantage. As you can see, when an animal is put into a hypotonic solution, it bursts. Whereas, plants have cell walls, so the cell just bulges. It doesn't burst. The bacterial cell wall has a little more to it. Now the bacterial cell wall is enclosed by a cell envelope. Due to the differences in the cell envelope or cell wall, bacteria is classified into two groups. One is the gram-negative bacteria containing thin peptidoglycan and the other gram-positive bacteria containing thick peptidoglycan. Due to the difference in the composition of blood calyx, bacteria can have a slimy layer or a capsule. In this picture, it's a capsule. Then chloroplasts, which are found in plants and algae. I'll tell you an easy way to remember these terms. But firstly, plastids, a double membrane organelle consists of different organelle pigments. Chloroplasts being one of the larger group of organelles containing chlorophyll. The site of photosynthesis and ATP synthesis. Chromoplasts contain carotene as such, providing a yellow, orange or red color to the plants or to the fruits when it ripens. Leucoplasts store nutrients, like amyloplasts store starch. Now let's look at these terms. So each chloroplast has a double membrane. The outer layer and the inner layer. This inner layer forms branching membrane called thylakoids. These thylakoids are arranged in stacks like flattened discs called grana. Grana is the plural form for many stacks. And this thylakoids is the location of chlorophylls and pigments. Also, the fluid outside the thylakoid is called stroma, which contains chloroplast DNA and ribosome 70S, together with many enzymes. This is the chloroplast structure. This sac is the granum and the many sacs are grana. This fluid outside the thylakoid is stroma. This membrane is the thylakoid. Ribosomes too. And this interconnecting link joining different grana is the stroma lamella. The outer membrane and the inner membrane. Tonoplast, found only in plant cells. They are a specialized membrane surrounding the vacuole, acting as a barrier between cytoplasm and the fluid contents in the vacuole. Okay, lastly, additional features of organelles in animals and bacteria. We have the flagella or cilia for overall cell movement. The bacteria has three structures. Like villi, an extension which helps the bacteria to attach to other bacteria or host cells. Plasmid, they have genes in them providing antibiotic resistance. And the capsule, what we discussed earlier. This is to cause disease and protect the bacteria from being engulfed. Example is Salmonella. These are bacteria. Here's an overview on cellular process like growth and metabolism, DNA replication and protein synthesis, which are important for cells' activity and motility where cells move as long as they have flagella and cilia. The DNA replication. Eukaryotes undergo nuclear division, meiosis, the haploid during sexual reproduction, and mitosis, the diploid during asexual, and for cells' growth and replacement. Here you can see the processes. Prokaryotes are the simplest ones. This is the most simplified chart showing the differences between prokaryotes and eukaryotes. Take a note on the DNA, the synthesis and the organization. And lastly, viruses. These are non-cellular structures, active only in the host cells. They aren't living, and RNA and retrovirus are some examples. Alright, to observe cells, microscopes are used, and electron microscope produces a sharper image than light microscope. And use this triangle method to solve any kinds of cell calculations. That's it. We have come to the end, and I apologize if I went too fast in some areas. But thank you so much for watching. Hope you had fun learning all about cells.